Thank you so much for giving me the opportunity to talk. Today, I'll be talking to you about my work towards engineering cell-specific protein delivery vehicles for erythroid lineage cells. Sorry. And a lot of our work is, thinking about how do we expand this undruggable proteome. 85% of proteins associated with disease are considered undruggable, particularly because they work through these shallow protein-protein interactions that are intractable to small molecules. Yet, we do see that protein biologics, both naturally and engineered, can start to be versatile binders of these non-ligandable targets. By this, I mean antibodies, their mimics like nanobodies, designer peptides, as well as cyclic peptides. Altogether, my PhD work has been focused on thinking not only designing these binders for undruggable proteins, but also thinking about drug delivery systems that can cross the cellular membrane and help hopefully increase the scope of targets we go after. In particular, we're interested in targets that are related to sickle cell disease, as we think that transcriptional reprogramming is a major approach of therapeutic development. So sickle cell disease, as many of you may be familiar with, is a result of a single point mutation that can result in a polymerization whenever your red blood cells drop off oxygen at tissues. De-repression of fetal hemoglobin, which is an alternative hemoglobin protein, is sufficient for protective effect. And we actually know that clinically there's newly approved CRISPR technologies in which you can edit cells of interest to reduce the levels of the major silencer, which is BC11A. By doing such, you're actually able to induce higher levels of fetal hemoglobin, which ends up giving you that protective effect. And while this is potentially curative for sickle cell disease, we do still need to think about non-genetic therapeutic intervention for accessibility at a global scale. Past just that, BC11A is really interesting. It's highly disordered, so most of the protein cannot be structurally resolved. It's located inside of the nucleus, so if we think about the binders that we use, they do have to cross multiple membranes. And finally, it is widely expressed and plays vital roles across maturations of multiple stem cells. And so, altogether, we really establish a platform in which we've been able to identify binders, nanobodies specifically, that can bind with high specificity both in the ordered and disordered regions of BC11A. We've been able to engineer cell permeance, and that's what I'll be talking about mostly today. And finally, we can take this cell-permeable protein degrader in order to for targeted degradation of BC11A, recapitulate fetal hemoglobin, and to kind of start to build the basis of biomolecules for sickle cell disease. As mentioned, I'll be focusing primarily on designing and engineering cell permeance, and I'm hopefully by the end of this talk, if you wanted to deliver binders to your favorite protein inside of the cell, you have some ideas on how to do that. And so that's really the goal of my talk today. So there's a wide variety of mechanisms in which proteins can naturally enter the cell. So charge-induced transport, in which positively charged proteins can induce endosomal uptake and escape. For this, I'll be investigating two major players within the CPP, cell-penetrating peptide field, which are ZF5.3 and E5-TAT. We can also think about thiol activation at the cell surface, in which there are mechanisms of dilation that allows for direct permeability into the cell. And so for this, we'll be using TMBR10 as a model cargo. This is a sort of hybrid approach where it's both a CPP, but because it's activated, you can actually start to see the improvements of thiol activation over just charge-induced transport. And finally, I did also investigate receptor-mediated entry via bacterial toxins. I'm not going to be talking about that, but if anybody's interested, definitely happy to answer any of those questions. So altogether, the goal is how can we re-engineer these for delivery into erythrocytes, because those are their cells of interest in sickle cell disease. So first, we were able to establish that by binding ZF5.3 to the nanobody that binds to BC11A, we can get it inside of the cells in a dose-dependent manner, as well as it seems to be accumulating into the nucleus, which is preferred because our target is in the nucleus. And while this was excited and this was sufficient to deliver to show active degradation of BCL11A, it was a 10 micromolar, as well as highly unstable. And so really when you started to append various other ligands, you start to lose that stability. And so I wondered if not only can we improve the delivery, but we can also start to target it specifically to developing erythrocytes that are pretty hard to get proteins inside, specifically because complex spectrum networks right underneath the membrane is important for its integrity. So it's really hard to be able to get material inside of these cells. So I'm investigating uptake efficiency as well as trying to engineer the selectivity. In order to do that, I'm using our model cargo of neon green, which is just fluorescent, so it allows us to do both live cell confocal imaging, quantifying that, as well as flow cytometry. We're appending the vehicles I mentioned earlier, ZF5.3, E5 TAT, as well as 2 different conditions of our thiol-reactive CPP additives. The reason why I chose 2 different conditions is because it's thought that by having them in excess, you can actually get more delivery of your protein inside of the cells. So I'm also testing this in 2 different cell lines because we do believe erythrocytes are harder to access. And so altogether, by using these orthogonal techniques, we can compare total uptake and investigate inherent cell types differences across these vehicles. To summarize a lot of data, I'm going to just show you the median uptake. And so right off the bat, we're seeing the ZF5.3 shows the highest uptake. Great, we were able to use that before, so that kind of makes sense. We really see drop-off across these other vehicles. And interestingly enough, we are seeing that almost all of them show lower uptake into erythrocytes. And so really this motivates our next question, which is, can we actually engineer improved uptake? I predict that by using receptor targeting, we can increase the relative concentration at the cell surface, and this will allow us to improve uptake as well as engineer specificity in a mixed cell model. But which receptor do we use? Across erythropoiesis, there's many receptors that are highly expressed. In particular, we're focused during hemoglobin synthesis because that's when we're intervening. And across these 3 major ones, which are glycophorin A, erythropoietin receptor, and CD71, which is a transferrin receptor, the cleanest profile is with glycophorin A. And so we decided to move forward with using that. And after confirming that there's differential expression on the surface of the cells we're using, we also utilized a nanobody that has pretty good affinity to the membrane. And we wanted to see if just appending the ligand itself can improve that cell surface binding. And we did see some improvement, but not what we were expecting with a 200-fold difference in expression. But it was significant enough that we were like, let's move forward and see what are the results of appending this ligand, this targeting ligand, onto all of our delivery vehicles. So once again, we designed these proteins, purified them, and this is our results for our GERCAT cells. And what I'm showing here in red are the values without IH4 appended. And as you can see, ZF5.3 loses a lot of its ability to uptake. We see no differences within the TMBR10. And although E5 TAT looks like it's increased, a lot of that is due to a lot of aggregation outside of the cell. So it's greatly destabilized. And we see similar results in HUDEP2 cells, which are a erythroid lineage cell model, but we do actually see that specifically with the TNBR10s, we do get this synergy between receptor targeting and the cells we're trying to improve into. And so to kind of summarize that, not everything you add your receptor targeting to is gonna help. But specifically with our CPP additives, at least in single cell culture, we're seeing this benefit. So finally, the real question is does this make sense in a mixed cell model? So we did this mixing our cell types we co-cultured them, delivered our ligand, and then we were able to use a CD36 marker to differentiate between JERCAT and HUDEP. And as you can see here, we're actually seeing greater uptake into the HUDEP2 cells in this mixed co-culture. And when we quantify these images, we're actually seeing that's about a 2 to 3-fold increase, which matches the surface binding that we're seeing. So not only are we seeing synergy between these this specific CPP additives and adding the receptor, but the affinity to the cell surface is going to be the major kind of contributor to how much of an improvement you're seeing. We also use flow cytometry to analyze what are the concentrations we can use, and we're seeing this benefit in as little as 300 nanomolar. And our previous construct was delivered at 10 micromolar. So it does seem like this is beneficial. And finally, to put it all together, not only are we seeing the benefit in single cell culture, but also in mixed cell culture. And so when you're thinking about designing this for your own constructs, if you do want to get cell specificity, these CPP additives seem to have that kind of the ability to be engineered for that. But we do also know that these other CPPs do show generally more robust delivery. And with that, I just want to acknowledge my lab and also some major collaborators that helped me really with the confocal quantifications. The work is up on AC Biochem AU, so if you are interested in getting more detail, seeing more of the images, I didn't want to bombard you guys with too much confocal. But yeah, and my funding sources. And yeah, I'll take any question at this point. Thank you.